Continuing on here in chapter 10, talking about those medications used in surgery. Same learning objectives. We've already talked about the classification system last time, and now just continuing understanding the different types of medications and what they're used for. All right, now we're coming into the anti-infectives. So we have antibiotics, antivirals, so just things that prevent infection, very important in surgery. So these are going to kill pathogens or hinder their growth. So not always killing them, but again, preventing infection. Pathogens are just substances that can cause disease. So they're microbes, and that includes bacteria, viruses, or other agents that are able to cause disease. The categories of these, we have antibacterials, they kill bacteria, antifungals, kill fungi, antivirals, you get it, viruses. And so these are grouped according to the action and their use. So first of all, just a little bit of micro here. So bacterial identification. So before we can determine an anti-infective, again, we need to know which category. Is it bacteria that we're preventing, we're treating? Is it a virus? Is it a fungus? Because they don't overlap. An antiviral doesn't help against bacteria, and antibiotics don't help against viruses. They don't help against fungi. So again, it's important to be able to identify. So how do we identify bacteria? There's a stain called a Gram stain, and this was developed by Hans Christian Gram, hence the name Gram stain, in the late 1800s, and it's based on the ability of the cell wall. So again, most bacteria are single-celled, but they have that thick cell wall on the outside to help protect them. So it's whether or not stain will stay. Now, the reason for that is because we have gram-positive and gram-negative cells. Gram-positive will be purple in a Gram stain, and gram-negative will be pink. The reason for that is that gram-positive have a very thick cell wall, and they don't have an outer membrane, so they're going to hold on to that stain, whereas the negative have a very thin cell wall and an extra membrane on the outside, so the stain and the dye can move across that and not get trapped. There are also bacteria called acid-fast, and these bind to bacteria that have this waxy cell wall. And there are only a few different types of bacteria that have them, and that is the Mycobacterium. So, for example, that's what causes tuberculosis and leprosy. Some other stains are spore stains to detect those endospores that are being formed by some bacteria, and flagella to talk about their movement. This is what those stains look like. So we have the Gram stains here. We have these dark purple coccyx shape, so it helps us see the shape, and also again the purple would indicate that Gram positive. Whereas here we have the Gram negative, and these are more rods here, so we can see their shape, but again the pink indicating that it's Gram negative. And then our acid fast, which are kind of this pinkish color here. Then we have our endospores, where we have these little green dots. Those are the endospores that are forming. And then finally our flagella that we can see on the flagellar stain. And just a quick review here about the difference between prokaryotes and eukaryotes. Again, now we're talking about antibiotics. So we're killing bacteria, which are all prokaryotes. They're very simplistic as far as the parts that they have. They have DNA not inside of a nucleus, it's just their long strand of DNA in one little compartment. It's all kind of grouped together there. But again, all cells have DNA, they just don't have a nucleus. They have ribosomes, which is where proteins are made. They have cytoplasm here, which is just a fluid inside. And then they have a cell membrane that separates the inside and outside of the cell. But then whether they're Gram positive or Gram negative, what the cell wall, that outer layering past the cell membrane looks like, can be a little bit different. Again, just a quick review of those. The Gram negatives have a thin cell wall, and then they have another membrane called the outer membrane. And they also have that lipopolysaccharide on them that helps detect them. Whereas the Gram positive, they're going to have a thick cell wall and just the one membrane, and they have that teichoic acid that kind of differentiates them a little bit. So again, the antibiotics we're talking about, they're targeting these prokaryotic cells. Whereas eukaryotic, which are the cells that we have, and the whole goal of an antibiotic or any drug is to target like the microbe or the pathogen without harming our cells. So trying to find those differences. So again, we have DNA as well, but it's stored inside of a nucleus, so it provides an extra layer to protect it. We have ribosomes to make proteins, but a lot of antibiotics target those ribosomes to prevent protein synthesis. The reason that can happen is because the ribosomes of a prokaryotic cell are much smaller than eukaryotic cells. So antibiotics can specifically target the smaller ribosomes so that bacterial cells can't go through protein synthesis, but we still can. Again, cell walls are only on prokaryotics, so targeting that thin or thick cell wall made up of a carbohydrate called peptidoglycan is a nice target, whereas eukaryotes, like us, we don't have a cell wall, so it doesn't hurt us if you're damaging cell walls. So again, we also have organelles, mitochondria, and other compartments as well that prokaryotic cells don't have. All right, so if we look at these antibiotics, the first class we'll talk about is called the beta-lactams. These are antibacterial. Again, they're only going to help against some bacteria, not all of them. But this includes penicillins, the cephalosporins, and the carbapenems. So penicillin, one of the first antibiotics that was discovered completely by accident. So these, again, are not effective against the resting bacteria. So the only time that this particular group works is if the bacteria are actually actively multiplying. So if it's already established, these won't hurt them, but it will prevent them from producing other bacteria. So it inhibits the cell wall it inhibits an enzyme that makes the cell wall. So that's why it doesn't help with resting, because once the cell wall is made, it doesn't hurt it, but it prevents cells from making that cell wall. So again, penicillin, kind of our first antibiotic, it was actually made from a mold, again by mistake. Scientists was studying a bacteria, left his plates of bacteria out, came back and there was a mold growing, but then noticed that, oh, hey, around this mold there's a clearing of this bacteria. It must be able to make something that kills the bacteria. And then that led to the production and the history kind of our antibiotics. There are 4 classes of penicillins. So we have ampicillin-like, broad-spectrum, penicillin G-like, and penicillin ACE resistant. And so they all kind of work the same way. They're all inhibiting the production of enzymes. Broad spectrum means that they're affecting a lot of different categories, whereas narrow spectrum, just 1 or 2 specific types of bacteria. But typically, if somebody is allergic to one, which is common, they're allergic to all of them. The cephalosporins also came from a fungus that's found in soil. So it was used to be called cephalosporinium, which was what the fungus was called. Again, it's changed its name now, but that's the kind of history behind the naming of these. But again, the fungus was producing this enzyme to help protect it. It was kind of its immune system against bacteria and getting infected. And then we are able to mimic that to inhibit some bacteria. So quite a few classifications. They all start with ceph, so cephalosporin, ceph. So generations. So first generation just means the very first one in this category. So it was only effective against gram-positive cocci. Then, did some modifications. Now the second generation came out. Now we still have those gram-positive cocci, but now some gram-negative bacilli. Then a new generation, the third, specific types of bacteria, so narrow spectrum. Then the 4th generation, again, just some more of the gram-positive cocci, gram-negative bacilli. And then the 5th generation helps against MRSA, which is the methicillin-resistant Staph aureus. So again, as we're modifying and kind of honing in on these different antibiotics, they have different generations, so they're effective against different things. The carbapenems, this came from Phenomycin, again, which is actually a bacteria that's found in soil. And so you can see Catalina in there, so it was found on farms. So instead of coming from a fungus, it actually comes from another bacteria that is producing an enzyme that kills the other bacteria. So it's a competitive mechanism. So this can be used to treat Haemophilus influenza, some anaerobes, meaning bacteria that don't like oxygen, and the Enterobacteriaceae, which are a lot of the things that in our GI system that cause problems, and then that methicillin-resistant Staph aureus, the MRSA. Couple examples here, Intrapinum and Imipenem. Our next category here are the sulfonamides. Now this is a synthetic, so the other ones came from, again, they were natural in that they came from other living things, again, bacteria or fungi. Now the sulfonamides, this is completely synthetic, meaning completely man-made by putting chemicals together. It disrupts folic acid synthesis, which is unique to prokaryotic cells. We can't make folic acid, so inhibiting this pathway doesn't inhibit our cells, but it does inhibit different bacteria. So this helps with the gram-positive and gram-negative, both classes. But again, lots of allergies to this as well as resistance, meaning that the bacteria that used to be, harmed by these drugs now are resistant. And just a few examples here. Sulfacetamine, so you can see they all start with sulfa methoxyolone, and then sulfasalazine. All right, now our polypeptides here are other categories. So these are also called polymyxins. Again, back to kind of a more natural. So this is made by a bacteria called Bacillus polymyxia, hence the name. Now these bind to the cell membrane and prevent the release of toxins. Okay, so this would be helpful against the type of bacteria that is making toxins as part of its virulence. This doesn't actually kill the bacteria, but it prevents the release of the toxin, so it makes it much less harmful. So it's good against the gram-negative bacteria. Some examples here, bacitracin, polymyxin, and B and E. A lot of times these can be found in some of our neosporins and some topical treatments. So again, it's helping to prevent the release of those toxins. And just a quick note here about these. I'm mainly concerned that you understand how these different categories work. I'm not going to ask specific examples like what is polymyxin, which category does it go into, but I want you to know that polypeptides are a type of antibacterial. They harm bacteria, and specifically they're preventing the release of toxins. Those are the things that I want you to concentrate on. All right, the next category are the aminoglycosides. So these are, again, natural. They're derived from a couple different microbes, Streptomyces and Micromonospora. So myosin comes from Streptomyces, and then mycin from the Micromonosporium. So these interrupt the protein synthesis. So again, back to those ribosomes that are making proteins. So by inhibiting the production of proteins, the cells are not going to be able to grow or survive. Some of these can also degrade the cell walls. And again, this is degrading them. So the penicillins prevent the synthesis. So not good against something already established, but these could actually help because they're actually breaking down the cell wall that's already there, which kills the cell. And these treat a few different things like the gram-negative, aerobic, meaning they like and need oxygen, versus the anaerobes that don't like any oxygen, more in our gut. So the bacilli, and then facultative anaerobic, that just means that they prefer to have oxygen, but if oxygen is not available, they can switch to an anaerobic form, so fermentation, in order to get their energy. And then a few examples here, gentamicin, kanamycin, neomycin, again, neosporin has that, and streptomycin. All right, now our tetracyclines, again, a natural, so our only synthetic one that we've talked about are the sulfonamides or the sulfa drugs. So the tetracyclines come from acintobacteria, again, other bacteria that's producing chemicals to harm other bacteria. So it can outcompete them. This also disrupts the protein synthesis in order to stop the growth of that microbe. So this is good for a variety of gram-positive and gram-negative bacteria. A couple examples: doxycycline, minocycline, and tetracycline, hence the NIC category. All right, so then switching away from the antibacterials now to antifungals. Now, fungi are eukaryotic, so it can be a little trickier to treat them because, again, how do we harm the fungal microbe without harming the host that is also eukaryotic? These are also called antimycotics. So again, these treat fungal infections, which are also called mycoses. So things like fungi, mold, yeast, so like a yeast infection or athlete's foot, we would need an antifungal, not an antibacterial, because again, they have very different structures. So an antibiotic doesn't help against athlete's foot or something fungal, and an antifungal doesn't help against a bacterial infection. These work by inhibiting their growth or killing the fungi. So some of them are just slowing them so that they are slowed down so the immune system can get rid of them. Others actually kill the organism that is there. Three main classes of the antifungals: polyenes, azoles, and echinocandids. So our first one, the polyenes, these form channels in the fungal cell wall. So while our cells don't have a cell wall, fungi and plant cells do. Now, plant cells aren't harmful, so we don't talk about drugs to treat them, but fungi can be. So again, another nice target is the cell wall because we don't have cell walls. Now, that cell wall is different. It's made up of a carbohydrate called chitin instead of peptidoglycan from the antibiotics. So again, one doesn't help with the other. If you have a fungal infection, using penicillin that also impacts cell walls is not going to help because it's inhibiting bacterial cell walls, not fungal. So this prevents the reproduction. A couple of different examples: amphotericin, nystatin, and pimarisine. All right, now the azoles, these inhibit enzymes that form the structure of the cell walls and some of the internal functions. So again, that's a common place to target, is that fungal cell wall, because we don't have that. This is going to prevent the growth. So again, if it's already established, it's not going to be quite as helpful, but it will prevent it from spreading or from multiplying. A few examples, again, they all end with azole, so that makes it nice. So fluconazole, itraconazole, etc. Then our last category of the antifungals are the echinocandids, and these inhibit the cell wall synthesis. So again, we can poke holes in them. We can actually inhibit the enzyme that is needed for the structure, or we can just flat out prevent them from making it. So again, wouldn't help with an established infection, but can help with those that are reproducing. A few examples here, you can see they all end with fungin, so caspofungin, mycofungin, and digilafungin. All right, now if we look at some just general purpose antimicrobials, so now we're not getting antibiotics or antibacterials, just help with bacteria, antifungals only against fungi. Antimicrobials are used in surgical settings, so these would be antiseptics, disinfectants, and sterilants. So again, that's depending on whether or not they're making contact with the skin and the level to which they are destroying microbes. So we'll talk more about them in a minute. So the concentration and exposure time is actually what can differentiate these. So like alcohol, ethanol, for example, can be an antiseptic if it's diluted and it's just being applied for a little bit, like wiping down a stethoscope, or it can be a disinfectant if it's left on an area long enough for it to work. So that all of that is set by the CDC. So back to our policies and procedures. So this is what governs how long you should treat an instrument or how you should prepare a surface based on these categories. So the variations allow, again, the same agent to be effective at different levels. So again, alcohol, is a nice antiseptic, but again, if we leave it on longer, we use a higher concentration, it can now become a disinfectant. All right, so a couple different things here. So antiseptic, what does that even mean? So that means that it's used against microorganisms on living tissue. So again, alcohol on prepping before giving an injection. So you wipe the patient's arm or wherever with the alcohol. It is now an antiseptic because you are treating the microbes on something that's alive. A couple terms: biostasis is causing inactivity, so it's not killing it, it's just slowing it down. Biocide is actually killing it. Okay, speaking of killing, sterilants, these are chemicals or a process that kills all microbes, so completely eliminating things. So we have different chemicals that help, but also things like autoclaves or boiling or heating that can help with that as well. So sterilants also include spores. So again, a few of those like the bacillus and the Clostridium, they can produce those endospores, which are then released. And those endospores are extremely dormant. They can live outside of the host for extremely long time. So sterilizing something means that all of those are removed as well. And a few examples here: ethylene oxide gas, glutaraldehyde, peracetic acid, hydrogen peroxide, plasma, and the steam that is now available that they have. All right, so then our disinfectants. So these are used on fomites or inanimate objects. So if we're using them on tissues, that is a different term than what we're using for those on something inanimate, or not a term, which are disinfectants. So these are destroying those pathogens, and we have 3 different levels. So low level, intermediate, and high. So our first one, the low level disinfectants, this is going to kill most bacteria, some viruses, meaning they denature them, and some fungi. So this is not going to get rid of spores or things that are more resistant, like the mycobacteria that has that waxy cell wall, and that's what causes tuberculosis. It can be used on non-critical items, so those only contacting the skin. So again, using an alcohol wipe to wipe down a stethoscope, that's a non-critical item. It's not penetrating through the skin. So then that could be used in that scenario. So I already mentioned the alcohol here. So isopropyl alcohol, sodium hypochlorite, phenol, iodophors, and quaternary ammonium. Now the intermediate, again, this could be the same molecule, it's just being used for longer. So this kills most bacteria, fungi, and viruses. So not everything, but it does kill a lot of pathogens. Again, the exception are the spores. So these are going to be used on semi-critical, just meaning they're making contact, but they also can come into non-intact skin and mucous membrane. So maybe just like a small scrape or something, but now you've opened up those underlying areas to the potential for microbes. A few examples here again: the alcohol, sodium hypochlorite, phenol, and the iodophor, like iodine. Again, concentrations and exposure are increased in order to move from the low level to the intermediate level. Now our high level, our last one for this category or for this part of the chapter 10. So high level is actually going to sterilize things. It's going to kill all microbes. However, it does not get rid of the spores or the prions. So we do need autoclaves for that. So this will be used on those critical items so they come in contact with that sterile tissue. And then the sterilization is preferred, but some items are too delicate, so they can't undergo all these chemicals being on them. They start breaking down. So then depending on their use, the low level or intermediate level is preferred. And then just a couple examples here: glutaraldehyde and orthothethylaldehyde. Just a little tricky word there. So again, these are high level, so if we use them long enough at the right concentration, they can actually get rid of almost all microbes.